Hello dear students, in this lecture we are going to study the structure of prokaryotic cells. Before going in detail of the prokaryotic cells, we will first discuss about the shapes and the examples of some prokaryotic cells. Bacteria like E. coli, cyanobacteria which are also called as blue-green algae, mycoplasma, PPLOs are Pneumonia-like organisms. These are some examples of prokaryotes. Mycoplasmas and PPLOs, they are wall-less microorganisms, and bacteria and cyanobacteria, they possess the cell walls. Then these prokaryotic cells in general, they are very small in their size as compared to the eukaryotic cells. And the prokaryotic cells divide faster than the eukaryotic cells. For example, a typical eukaryote E. coli cell which has favorable growth conditions can divide every 20 minutes. And in comparison to that, the eukaryotic cells take very long time to divide. For example, typical human cells in cell culture in the laboratory, they take 24 hours for one cell division, but E. coli can divide every 20 minutes. So because they are smaller and their generation time is also small, they divide very fast and they produce large number of cells within less amount of time. Then there are some types of prokaryotic cells based on their different shapes. So 4 different types of shapes or types of cells are generally observed in the prokaryotes. The first type is coccus, which is a spherical shape of the bacteria, and the shape is something like this. The cocci can be isolated like this, or they can be present in the form of a strip like this. And if they are present in the form of a strip like this, then they are called as Streptococcus. Okay, so Streptococcus is one of the genus of the bacteria, and this is one example, Streptococcus pneumoniae, which causes pneumonia in mammals. So this is just one example of the bacteria having the coccus type of cell shape. Then the second type of the group of the bacteria having a rod-like shape is Bacillus, and the example of Bacillus or rod-shaped bacteria is Bacillus Thuringiensis, which we will study in the 12th standard in detail. And this bacterium or its genetic material, small part of its genetic material, is actually used in the production of BT cotton that we use in these days. Then the third type of the shape in case of bacteria is Vibrio, which is a comma shape like this, and the example of such type of bacteria is Vibrio cholerae, which causes cholera in humans. And the fourth type of a typical shape, a common shape observed in bacteria is spirillum, something like this, a spiral-like structure. And the example of such type of bacteria having spiral type structure is S. volutens. S indicates the name of the genus Spirillum, and volutens is the name of the species. So these are different shapes of the bacteria, and I have given one example of each shape, but there are n number of bacteria, actually trillions of bacteria or more than that also in the universe, and there are different type, n number of examples of each type of bacteria also. Okay, but these are in general the 4 different types of shapes which are generally observed in the prokaryotic cells. Now we will come to the actual structure of a typical prokaryotic cell. For example, this is the structure of a bacterial cell, and we can observe that there are different types of parts of a bacterial cell. अब आपके NCERT के सिलेबस में ऑर्गेनाइजेशन ऑफ द सेल में हर पार्ट के बारे में एक सेंटेंस दिया हुआ है, एंड देन अगेन दे हैव एक्सप्लेन्ड एवरी पार्ट इन डिटेल. सो वी विल फ्रॉम फर्स्ट डिस्कस एवरी पार्ट इन डिटेल, तो 2 बार उसको सेम चीज को पढ़ने की जरूरत नहीं होगी. तो फ्रॉम आउटसाइड टू इनसाइड, what are different parts of a prokaryotic cell that we will see here. The outermost protective covering of the prokaryotic cell is called as cell envelope. and this cell envelope has different modifications in different types of bacteria. And this cell envelope is made up of 3 layers. The outermost layer of the cell envelope is Glycocalyx, but this Glycocalyx is not present in every bacteria. It may be present in some bacteria, it may not be present in others. Okay, and this Glycocalyx is made up of generally it is made up of polysaccharides or some oligosaccharides, matlab some carbohydrates, but the actual composition of the polysaccharides or the oligosaccharides in the Glycocalyx differs from bacteria to bacteria. And this Glycocalyx, which is made up of carbohydrates or polysaccharides, it may be thin in some bacteria, मतलब कुछ बैक्टीरिया में प्रेजेंट होगा, कुछ में नहीं होगा, और जिनमें प्रेजेंट है उनमें उसका एक तो thin layer हो सकता है, thin and loose layer, and in that case the layer is called as slime layer. And if the Glycocalyx is very thick and tough, then it is called as capsule. And capsulated bacteria, they are very resistant to the immune systems of the mammalian bodies, and they are actually more virulent or dangerous for the human beings and other mammals. So these capsules or these slime layers, they protect the bacteria from the immune system of their host cells, जिन host को वो infect करते हैं. For example, Streptococcus pneumoniae के 2 different strains होते हैं. एक strain मतलब variant, तो एक वेरिएंट ऐसा होता है कि जिसके ऊपर कोई कैप्सूल मतलब ग्लाइकोकैलिक्स नहीं होता है, तो वो रफ दिखाई देता है अंडर द माइक्रोस्कोप, इसलिए हम उसको रफ स्ट्रेन बोलते हैं. सो दैट स्ट्रेन इज नॉट हार्मफुल फॉर द ह्यूमन बीइंग्स. इट कैन नॉट कॉज न्यूमोनिया इन द ह्यूमन बीइंग्स और इन द मैमल्स. बट another strain which is having the glycocalyx in the form of capsule is resistant to the defense mechanism of mammals or human beings, and that is why it can cause pneumonia in different types of mammals. Okay, so in general this glycocalyx in the form of capsule generally protects the bacteria from the immune system of the host cell, but it is also involved in the protection of the bacteria from the mechanical damage also. Okay, in addition to the glycocalyx, the second layer, protective layer that is present beneath the glycocalyx is cell wall. Okay, and the cell wall is also made up of different types of polysaccharides. The most important polysaccharide that is present in gram positive bacteria is peptidoglycan. So we'll see the details of the cell wall here. Now cell wall in case of gram positive bacteria is made up of peptidoglycan, which is a complex polysaccharide, and in case of gram negative bacteria it is made up of murine and muramic acid. Or N-acetyl muramic acid. So what is exactly gram positive and gram negative, that we will discuss here. This distinction between the gram positive and gram negative bacteria is basically based on a staining procedure. मतलब माइक्रोस्कोप के अंडर जब हमको बैक्टीरिया को ऑब्जर्व करना होता है तो वी जनरली स्टेन और कलर द बैक्टीरिया, एंड देन वी सी डिफरेंट कलर्ड पार्ट्स ऑफ द सेल अंडर द माइक्रोस्कोप. तो कुछ पार्ट्स कलर लेते हैं इसलिए वो मतलब आसानी से दिखाई दिए जा सकते हैं अंडर द माइक्रोस्कोप. इसलिए हम जनरली बिफोर द ऑब्जर्विंग एनी सेल अंडर द माइक्रोस्कोप वी स्टेन देम, वी कलर देम, एंड such type of one stain was used by a scientist called as Gram, and he named that stain as Gram stain. So what he did, gram stain is basically, he prepared a gram stain which is basically an alkaline solution of crystal violet. So this crystal violet is a stain or a coloring agent, और उस स्टेन का या कलरिंग एजेंट का अल्कलाइन सॉल्यूशन, वीकली अल्कलाइन सॉल्यूशन इस साइंटिस्ट ने यूज किया था टू कलर और टू स्टेन द बैक्टीरियल सेल्स एंड देन टू ऑब्जर्व देम अंडर द माइक्रोस्कोप. तो इसी स्टेन को उसने खुद का नाम दे दिया, दैट इज ग्राम स्टेन. सो द सेम स्टेन इज कॉल्ड एज ग्राम स्टेन. So what he did, he took different types of bacteria for observation. Suppose this is one type of bacteria which he took over the slide. एक स्लाइड ले ली, उस स्लाइड के ऊपर ये बैक्टीरिया ले लिए, फिर दूसरे स्लाइड के ऊपर कुछ अलग टाइप के बैक्टीरिया ले लिए, and then what he did, he heat fixed these bacteria on the slide. मतलब स्लाइड के ऊपर से बैक्टीरियल सस्पेंशन डाला हुआ है, लिक्विड फॉर्म में बैक्टीरिया प्रेजेंट है लिक्विड सस्पेंशन में, एंड स्लाइड के नीचे से हीट दे दी ओवर द बर्नर. ओके, सो दैट इज कॉल्ड एज हीट फिक्सेशन. सो ड्यू टू हाई टेम्परेचर ऑफ द स्लाइड, ड्यू टू द फ्लेम अंडर द स्लाइड, द बैक्टीरिया विल बी किल्ड एंड दे विल बी फिक्स्ड ऑन द स्लाइड. दैट इज कॉल्ड एज हीट फिक्सेशन. सो he performed heat fixation that applies to both the slides. दोनों स्लाइड के ऊपर बैक्टीरिया हीट फिक्स किए, and then he applied the gram stain over the two slides. Next step is addition of gram stain. Okay, after adding the gram stain, he added a mordant which actually gives strength to the gram stain, so that the gram stain can very firmly adhere to the cellular parts. But we won't write that mordant step here. You just remember that a mordant is added. Mordant is an iodine solution which is added for the better adherement of the gram stain over the cell wall of the bacteria. And then he added a decolorizing agent in the form of an alcohol. Okay, so alcohol डालने से decolorization होता है, मतलब cells can lose the stain. तो जो colored stains है, cells है, they can become colorless again. So gram staining के बाद उसने observe किया, तो the cells were looking purple under the microscope. These cells were also looking purple, these cells were also looking purple. Okay, but decolorizing agent डाल दिया, तो decolorizing agent डालने के बाद इन सेल से तो स्टेन निकल गया, बट इन सेल से स्टेन नहीं निकला, क्योंकि decolorization हर सेल का भी नहीं होता है. कुछ सेल्स स्टेन को बहुत फर्मली पकड़ लेते हैं ड्यू टू ए स्पेशल टाइप ऑफ कंपोजिशन प्रेजेंट इन देयर सेल वॉल, एंड कुछ सेल्स उसको फर्मली नहीं पकड़ पाते बिकॉज दे डोंट हैव दोस कंपोनेंट्स इन देयर सेल वॉल. तो अल्कोहल से इन सेल्स का डीकलरेशन हो गया, इनका नहीं हो गया. सो दे आर स्टिल पर्पल इन कलर बट दे आर कलरलेस. ओके, एंड आफ्टर decolorization step, the scientist Graham added second stain which is safranin. So safranin will be accepted by only those cells which have already lost the gram stain, but these cells which have retained the gram stain, they won't accept the safranin, and that is why even after addition of the safranin they will look purple in color, but they won't look purple, they will look red in color. Okay, so based on this, which cells retain the gram stain and which cells don't retain the gram stain, gram scientists named bacteria as gram positive and gram negative. So जिन cells ने gram stain लास्ट तक नहीं छोड़ा, retain करके रखा even after the addition of decolorizing agent, that is alcohol, they are called as gram positive. So these are called as gram positive cells. एंड जिन सेल्स ने डीकलराइजिंग एजेंट मतलब अल्कोहल डालने के बाद ग्राम स्टेन लूज किया और फिर उन्होंने दूसरा सैफ्राविन स्टेन एक्सेप्ट किया, उन सेल्स को हम कहते हैं ग्राम नेगेटिव सेल्स. ओके, सो बेस्ड ऑन द स्टेनिंग प्रोसेस we say bacteria as gram positive or gram negative. But what is that chemical component in the cell or cell wall which is responsible for retaining the gram stain? That component is basically peptidoglycan. So जो बैक्टीरिया ग्राम पॉजिटिव होते हैं उनके सेल वॉल के अंदर बहुत ज्यादा पेप्टाइडोग्लाइकन होता है, मतलब दे हैव ए थिक लेयर ऑफ द पेप्टाइडोग्लाइकन एंड दिस पेप्टाइडोग्लाइकन captures and retains the gram stain, that is weak alkaline solution of crystal violet. They don't lose it even after decolorization, and that is the mechanism why they appear purple even after the addition of alcohol and safranin. Okay, and these cells which lose the gram stain, they actually don't have that much amount of peptidoglycan, or they not at all have peptidoglycan. मतलब they may have a very little amount or they may not have any amount of peptidoglycan in their cell wall, and that is why they very easily lose the gram stain and are called as gram negative bacteria. So this is about the nature of the cell wall in bacteria. But this cell wall, functionally it is very important, whether it is gram positive or gram negative cell wall. It is very important for the bacteria because bacteria don't possess cytoskeleton which is present in eukaryotic cells, and that is why to provide shape and structural rigidity to the bacterial cell wall is the only important component. Okay, so cell wall provides proper shape to the bacteria and it also provides structural rigidity to the bacterial cell so that the bacterial cell does not collapse or it doesn't get burst. Okay, and the cell wall also protects the bacteria from mechanical damages also. The third layer of the cell envelope is plasma membrane, and plasma membrane is made up of lipids and proteins. Structurally, plasma membrane is similar, the bacterial plasma membrane is similar to the eukaryotic cells plasma membrane. Both the plasma membranes, matlab plasma membrane of any cell, whether they are bacterial cells or eukaryotic cells, the plasma membrane is made up of lipid bilayer having proteins embedded in it. We will see the structure of plasma membrane later on. तो जो structure हम बाद में देखेंगे, the same structure applies to the bacterial plasma membrane and the eukaryotic plasma membrane. The types of the lipids and proteins in the plasma membrane may differ, but the actual architecture or the structure of the plasma membrane is similar in both the cases. सो ये लिपिड बाइलेयर का बना हुआ है जिसमें प्रोटीन से एम्बेडेड होते हैं, तो फ्लूइड मोज़ेक मॉडल इसको हम कहते हैं प्लाज्मा मेम्ब्रेन को. वी विल डिस्कस अबाउट इट लेटर ऑन जब उसका टर्न आएगा तब. बट ये बैक्टीरियल प्लाज्मा मेम्ब्रेन जो होता है इट इज वेरी इंपॉर्टेंट एंड इट परफॉर्म्स डिफरेंट टाइप्स ऑफ फंक्शंस. द बैक्टीरियल प्लाज्मा मेम्ब्रेन हैव सम इनवेजिनेशंस, बहुत सारे इनवेजिनेशंस मतलब इनफोल्डिंग्स होते हैं इस तरीके के इनटू द साइटोप्लाज्म फ्रॉम द प्लाज्मा मेम्ब्रेन, लेकिन यहाँ पे सिंप्लिसिटी के लिए मैंने सिर्फ एक इनफोल्डिंग दिखाया है. एंड सच टाइप्स ऑफ इनफोल्डिंग्स और इनवेजिनेशंस ऑफ प्लाज्मा मेम्ब्रेन इनटू द साइटोप्लाज्म आर कॉल्ड एज मेसोसोम्स. एंड these mesosomes they increase the surface area of the plasma membrane and this helps bacteria a lot क्योंकि प्लाज्मा मेम्ब्रेन के ऊपर बहुत सारे एंजाइम्स होते हैं. बैक्टीरिया के अंदर माइटोकॉन्ड्रिया नहीं होता है, इसलिए रेस्पिरेटरी एंजाइम्स जो होते हैं सेलुलर रेस्पिरेशन वाले एंजाइम्स दे आर प्रेजेंट ऑन द प्लाज्मा मेम्ब्रेन एंड ड्यू टू द प्रेजेंस ऑफ दिस मेसोसोम्स द सरफेस एरिया ऑफ द प्लाज्मा मेम्ब्रेन increases, which increases the content or amount of the enzymes, respiratory enzymes present on the plasma membrane. But in addition to the respiratory enzymes, the plasma membrane or the mesosomes, they also contain other different types of enzymes for different types of activities. Okay, another function of the mesosomes is that they get attached with the bacterial chromosome which is present in the form of a nucleoid. We know that in case of prokaryotes and in bacteria there is no nuclear envelope surrounding the chromosome or the DNA, and that is why we don't call it as nucleus. So the DNA जो होता है बैक्टीरिया का वो एक सिंगल सर्कुलर डबल स्टैंडर्ड डीएनए होता है, मतलब एक ही क्रोमोसोम होता है बैक्टीरिया के अंदर जो सर्कुलर होता है, डबल स्टैंडर्ड होता है, एंड सर्कुलर होने के बावजूद ये फोल्डेड भी होता है, इसमें बहुत सारे फोल्ड्स मतलब कॉइल्स एंड सुपर कॉइल्स भी होते हैं. एंड द रीजन वेयर दिस सिंगल सर्कुलर डबल स्टैंडर्ड डीएनए और क्रोमोसोम इज प्रेजेंट इज कॉल्ड एज न्यूक्लियोइड. एंड we were studying the second function of the mesosome. This mesosome provides an attachment site for the nucleoid, and it also helps in the replication, मतलब duplication of this DNA or chromosome and its distribution to the two daughter cells during the process of cell division. मतलब इस एक DNA से जब दो DNA बनेंगे, दो chromosomes बनेंगे, तो उन दोनों क्रोमोसोम का एक एक डॉटर सेल के लिए, मतलब ये सेल बाद में डिवाइड होगा बाय बाइनरी फिशन, तो एक सेल में एक क्रोमोसोम एंड दूसरे सेल में दूसरा क्रोमोसोम, ये पार्टिसिपेशन करने के लिए मतलब उनका डिस्ट्रीब्यूशन करने के लिए भी मेसोसोम हेल्प करता है. द थर्ड फंक्शन ऑफ मेसोसोम इज दैट दे आल्सो हेल्प इन द प्रोसेस ऑफ सिक्रीशन. सेल के बाहर अगर कुछ मतलब वेस्ट प्रोडक्ट्स डिस्चार्ज करने हैं तो दैट प्रोसेस इज सिक्रीशन और एक्सोसाइटोसिस, एंड the mesosomes also help in the process of secretion. The mesosomes or the plasma membrane, they also contain enzymes for the synthesis of cell wall components. So that is why we say that the mesosomes or the plasma membrane, they are also helpful in the synthesis of cell wall in bacteria. Okay, so these are different types of functions of mesosomes in the bacterial or prokaryotic cells. And in cyanobacteria, that is photosynthetic bacteria, there are some additional types of infoldings, and those infoldings contain photosynthetic pigments, and those types of infoldings or invaginations, they are called as chromatophores. Okay, so chromatophores contain the photosynthetic pigments in cyanobacteria or blue-green algae. Okay, so this is about the cell envelope, which is made up of glycocalyx, cell wall, plasma membrane. Glycocalyx is optional. It may be present in some bacteria, it may not be present. If it is present, it can be slime layer, a loose thin layer of the glycocalyx, or it can be a capsule, a thick and tough layer of the glycocalyx. But cell wall and plasma membrane, they are compulsorily present in bacteria. Cell wall may be made up of high amount of peptidoglycan or no or little amount of peptidoglycan, and based on that it is either gram positive or gram negative. And the plasma membrane has several invaginations in the form of mesosomes, and in cyanobacteria in the form of chromatophores also. This is about the cell envelope. We have to talk about the other structures of the bacterial cell also, but we will discuss about them in the next section. Thank you.